Hi viewers, in this class we are going to discuss recombinant DNA technology or RDNA technology and applications. Under this topic we are going to discuss introduction, steps involved in the recombinant DNA technology, tools of recombinant DNA technology, techniques used in the RDNA technology, and applications of RDNA technology. Let us start with the introduction. All organisms contain same four bases along backbone of DNA molecule, and number of bases present in them may vary. The information coded in DNA is transcribed into mRNA, further translated into protein, which are key molecules in carrying out all functions. A different protein can be made by changing DNA sequence. On this basis, a new technology called recombinant DNA technology has been evolved, and it was developed into the form of RDNA technology. RDNA is an artificial DNA formed by combining DNA fragment from two or more sources. It is also called as the chimeric DNA. Most commonly, RDNA involves introduction of fragment of DNA molecule from one organism to a second organism or another organism. It is prepared under in vitro conditions and introduced into organism with help of vector. Using RDNA technology, many human proteins have been synthesized in bacteria, yeast, or cultured mammalian cells. The RDNA experiment was first conducted by Boyer and Cohen in 1973. They devised techniques to isolate plasmids, cut them at precise locations, recombine the foreign DNA by ligase, and finally introduce them into another cell with precision, thereby creating a recombinant bacterium. In this way, bacterial cell produce desired proteins. See here, there are many number of recombinant human proteins, and they may be used to treat various diseases. Let us see some of them. Insulin, diabetes mellitus type 1, factor 8, blood clotting factor 8, hemophilia A, factor 9, hemophilia B, somatotropin hormone, growth disorders, tissue plasminogen activator, heart attack, adenosine deaminase, severe combined immunodeficiency. Like that, there are many kinds of proteins have been synthesized by using RDNA technology, and they may be used to treat many different diseases. See the basic principle of RDNA technology. Here there is a isolation of DNA, that is the DNA containing the gene of interest is isolated from the vector, and we go for the restriction digestion, that is restriction enzymes cut vector DNA at precise location, and foreign DNA which contain gene of interest by producing sticky ends or blunt ends. And then we will go for the ligation. The vector DNA fragment is ligated with foreign DNA, and introducing the RDNA into the desired cell, that is the bacterial cell. The RDNA molecule is introduced into the bacterial cell by transformation to produce the several copies of our desired DNA fragment, and screening of recombinants also will be done. That is, the cells containing several plasmid will grow into several colonies. To identify desired colonies, we screen them by replica plating and hybridization. So uh, here we are combining two different kinds of DNA. That is the basic principle behind the RDNA technology. So here we are using the isolation of target DNA and the isolation of vector, formation of DNA fragments, that is by using the restriction diation, cDNA synthesis or PCR chemical synthesis, ligation of cloning vectors, and screening of recombinants. Here there is a brief introduction where you can see this is the bacterial cell. We have isolated the plasmid from the bacteria, and we also isolated a desirable DNA from the eukaryotic cell or any cell of our interest, DNA isolation, and we have isolated a gene of our interest and inserted them into the plasmid here, and then this plasmid again sent into another bacterial cell here, and then the cells with the desired DNA fragment in the cloning vector, they will allow to multiply of our gene of our interest, and then we will make copies of our gene of interest, and they may be utilized in different fields. That gene may be the desired gene may be the gene may be gene for the pest resistance inserted into the plants. The gene may be used to alter bacteria for cleaning up toxic waste in the pollution-free environment, for the pollution-free environment, and the gene may be responsible for the synthesis of proteins used to dissolve the blood clots in the heart attack therapy, or the protein may be used to make snow from the higher temperatures. So this is the basic brief introduction to the RDNA technology and steps involved in the RDNA technology. Number 1, selection and isolation of DNA insert. Introduction of DNA insert into vector to form RDNA molecule. Recombinant DNA molecule is introduced into suitable host. Selection of transformed cells. Expression and amplification of DNA insert in the host. These are the basic steps that are involved in the RDNA technology. Let us see one by one. Isolation of DNA or selection and isolation of DNA of our interest. This is the first step in recombinant DNA technology, that is the selection of DNA segment of interest which is to be cloned. This desired DNA segment is then isolated enzymatically. This DNA segment of interest is termed as a DNA insert for foreign DNA or target DNA or cloned DNA. Then a suitable cloning vector is selected. In the next step of recombinant DNA technology, most used two vectors, they are the plasmids and the bacteriophages. So for the isolation of DNA insert, what we are doing is we have taken the suspension of cells in the suspension, and we have lysed the cells, and if you go for the acidification with the reagents, then there is a cell debris in the precipitate and DNA in the supernatant. This supernatant is transferred into the another vessel at the low pH. Here the DNA is adsorbed and wash the DNA with the buffer, and finally elute the DNA at the low salt concentration. So here we are taking out the DNA. Here also the same process. Cell suspension, lyse the cell with the lysozyme or detergent, and DNA precipitates at the interface, take out, and add alcohol. So the superficial one, the alcohol layer, and at the bottom cell debris. Then insert a glass rod and twirl gently. Pick up the DNA strands on the glass rod, transfer them into the another test tube. So the aqueous solution which is present in the test tube that contain the DNA. Then second step, insertion of DNA insert into the vector. That is introduction of the DNA insert into vector to form the recombinant DNA molecule. Here we are seeing, here this is the foreign DNA. We have taken out the edges by utilizing the restriction endonuclease enzyme. At the same time, by using the same enzymes, we are removing a fragment from the colonial vector, that is the plasmid. Then we have removed it, and now here we are constructing a new recombinant DNA. This is from the plasmid and this is from the foreign source, and by using ligase enzymes we are combined both the fragments. The larger one is the DNA from the plasmid, and this is the our desired DNA fragment. Now previously this is the only plasmid, but here the plasmid contain a foreign DNA of our interest, so that it is known as the recombinant DNA. Recombinant DNA. Then here also same thing. So digest both the eukaryotic DNA and the plasmid DNA with the same enzymes now to ligate them perfectly to form the recombinant plasmids. And then next step is the RDNA introduced into host cell or suitable host. Recombinant DNA molecule is introduced into suitable host. The formed recombinant DNA molecule is introduced into the host cell by different mechanisms, which include the transformation, or by microinjection method, or by electrophoresis method. Transfection method usually selected hosts or bacterial cells like the E. coli. So here the plasmid is introduced into the bacterial cell. So this is the normal E. coli of introduction. It is becoming transformed cell which contain, instead of normal plasmid, it contains a RDNA plasmid. And here we are visualizing the different kinds of methods. Here we are using the projectile gun to introduce the DNA coated tungsten molecules, and here this method is a transformation, and here it is a microinjection method, and we may use certain vectors to insert this DNA fragment. Now here this is the transgene, new gene from the foreign source. Here there are different methods: transformation, transfection, and electrophoresis to insert the recombinant DNA into the suitable host. And selection of the transformed cells. So although you insert many DNA inserts into many different cells, whether they are transformed or not, we should confirm it. So then thereby we will select the transformed cells. Transformed cells are the host cells which have taken up the recombinant DNA molecule. So in this, the what we will do, we will check out the transform the cells and separate them from the non-transformed cells by using the marker genes. Here we are observing, see, this is the cell which is transformed, and this is a non-transformed cell, and allow them to develop in the nutrient medium with a antibiotic, which is the ampicillin. Then after overnight of the growth, only ampicillin resistance colonies will be developed and remaining cells may be died off. Like that we will select the transformed cells. That indicates that Only the transformed cells can capable to show resistance against the ampicillin, but not the non-transformed cells here. Then transformation procedure here, the bacteria with the lag phase, you have cultured them, centrifuge them, resuspend them in the cold calcium chloride, again go for the centrifuge, resuspend in the small volume of cold calcium chloride, and then you will see the removal of plasmid DNA and add the appropriate broth, incubate to allow the gene expression, and then dilute them and plate on the selective medium. Then thereby only antibiotic resistance transformants will grow and remaining cells may die off. So this is the development of the colonies in the petri dish in a appropriate medium. Expression and amplification of DNA insert in the host. There is amplification of DNA insert in the host. The selected transformed cells that are expressing the gene of interest are amplified to produce sufficient number of copies of that gene. So here we are observing the recombinant plasmid underwent the transformation. So this is present in the new cell, and we allow them to develop to get many number of copies. That process is nothing but the amplification. We are enhancing the number of recombinant DNA copies within the suitable host. And once again we will recall steps in the recombinant DNA technology. Here we are using a gene of our interest is the insulin gene. This is the human cell DNA, and this is the insulin gene. We have taken out the gene of our interest in the form of insulin gene, and we have selected a suitable plasmid. Both of them isolated or otherwise excised by using restriction endonuclease enzymes, and thereby adding them by using ligases form the recombinant DNA and transform them into a bacteria. So this transformed bacterium which contain the insulin gene, so allow them to develop the recombinant bacteria again able to secrete human insulin. So we develop the bacterial colonies. All the bacteria which are present in these colonies have the ability to produce human insulin because they are carrying human insulin in their plasmids. Hence, they are able to produce the insulin of the human. Same. The first step is isolate the plasmid DNA and the human DNA. Cut both the DNA samples with the same restriction endonuclease enzyme. Mix the DNAs, they join by the base pairing. The products are recombinant plasmids, and many non-recombinant plasmids also may be present. They may be removed. Introduce the DNA to bacterial cells that have a mutation in their own LAGZ gene. Plate the bacterial agar containing ampicillin and X-gall. Incubate until colonies will grow. And then here you will get the recombinant DNA. So this is the bacterial clone. And the tools which are used in the recombinant DNA technology. The important biological tools for the recombinant DNA technology are the nucleases, ligases, DNA polymerase, vectors, and the suitable host organism. Nucleases. Those enzymes which have the ability to digest the nucleic acids are called as the nucleases. That is, they break the phosphodiester bonds of the RNA and as well as DNA. The nucleases that break RNA and DNA are called as the RNases and the DNases respectively. There are two types of nucleases based upon their activity. They are exonuclease and the endonucleases. Exonucleases remove the nucleotides from both the strands of the DNA from both the ends, and whereas the endonuclease that excises the DNA molecule in the middle rather than at the periphery. Here the exonuclease remove the nucleotides from the ends, and there are certain examples like the BAL31 E. coli exonuclease 3 lambda exonuclease. So BAL31 remove the nucleotides from both the strands of DNA. Exonuclease 3 remove from the third prime end of the nucleotides, and lambda exonuclease remove from the fifth prime endonucleotides. And here endonucleases, they cleave the internal nucleotides. They cleave the internal nucleotides at specific site called the recognition site or the recognition sequence, otherwise the restriction site or target site. So here, if the endonuclease, it cuts at the middle of the DNA, that is the endonuclease. If they cut at a precise site or recognition site or the restriction site, then they are called as the restriction endonuclease enzymes, otherwise restriction enzymes. Presence of restriction enzymes was first reported by W. Arbor in the year 1962. He found that when the DNA of bacteriophage was introduced into host bacterium, it was fragmented to small pieces. This led him to postulate the presence of restriction enzyme. The first true restriction enzyme was isolated in 1970s from E. coli by Michelson and Yuvan. Thousands of restriction endonucleases are identified. Few of them are palindromes. Restriction endonucleases are first called as the restriction enzymes as they restrict growth of viral DNA in the bacterium. A restriction enzyme recognizes a unique sequence. The number of cuts made in DNA from an organism is limited. A typical bacterial DNA molecule contains roughly 3 into 10 to the power of 6 base pairs, which cut DNA into few hundreds to thousand fragments. And here there is another restriction endonuclease enzyme, which is the HIND2, 1970, discovered by Kelly, Smith and Nothus. That is a Haemophilus influenza D2. Haemophilus influenza D2. And there are the different kinds of restriction endonucleases, namely type 1 to type 4, type 1 restriction endonuclease, type 2, type 3 and the type 2S. Type 2S restriction endonuclease. They are frequently used in cutting off DNA fragments. Here there are different kinds of restriction enzymes and from which bacteria they have been isolated, the source. For example, E. coli R5, E. coli RV, that is the Escherichia coli, ALU1, Arthrobacter luteus, BAMH1, Bacillus amyloliquefaciens, E. coli R1, Escherichia coli, E. coli R2, Escherichia coli, PST1, Providencia stewarti, SAL1, Streptomyces albus, BAMH1, Bacillus amyloliquefaciens, and the sequence which is identified by these bacteria is given here. Same, if you cut the DNA with the Bacillus amyloliquefaciens H1, it forms a cohesive end rather than the blunt end. Whereas if you observe here, HAE3 Haemophilus aegyptius, it forms the blunt end rather than the cohesive end. So based upon their ability, they may form cohesive ends or the blunt ends which are underwent ligation afterwards. And the end products of the restriction enzymes, that they may form the sticky ends otherwise cohesive ends, and they may also form the blunt ends based upon their ability. See here, these are the sticky ends or cohesive ends, and these are the blunt ends. Here, the cohesive ends, they are easily undergoing the sticky ends, they are easily undergoing the formation of recombinant DNA technology. Here we are using the E. coli R1 at both the ends, and thereby they cut the DNA fragment and forms the sticky ends, and finally they formed into recombinant DNA. And coming to the ligases. The ligase is also called as the molecular sutures, whereas the restriction endonuclease enzymes are called as the molecular scissors, which can cut. The ligases are the molecular suture or molecular glue. And Gellert, Lehman, Richardson discovered DNA ligase. The function of these enzymes is to join two fragments of DNA by synthesizing phosphodiester bond. They function to repair single-stranded nick in DNA, double helix, and in recombinant DNA technology, they are employed for sealing the gap between adjacent nucleotides. Here we require different kinds of. There are two examples given: E. coli DNA ligase and T4 DNA ligase. So E. coli DNA ligase requires magnesium 2 plus, NAD plus, which acts on the double-stranded DNA with cohesive ends. And T4 DNA ligase requires Mg2 plus, ATP, acts on double-stranded DNA with blunt ends. So here these two fragments are combined together with the DNA ligase to form a RDNA. And there are another enzymes which are called as the DNA polymerases. These are the enzymes which synthesize a new complementary DNA strand from existing DNA template or RNA template. Most used DNA polymerase is DNA polymerase 1, prepared from E. coli. The Kelno fragment of DNA polymerase 1 is employed to make producing ends double-stranded by extension of shorter strand. Another type of DNA polymerase is reverse transcriptase, which is also used in genetic engineering. Reverse transcriptase uses of RNA as a template for synthesizing a new strand called as a cDNA, complementary DNA. It is used in construction of cDNA libraries. Another type of DNA polymerase used in genetic engineering is TAQ polymerase, TAQ polymerase, which is used in polymerase chain reaction. It is used in the polymerase chain reaction, that is a PCR. Then coming to the different techniques which are employed, which are used in the RDNA technology, we will see briefly we will see the names. RDNA techniques employ different kinds of techniques, that is gel electrophoresis, blotting techniques, DNA sequencing methods, artificial gene synthesis, polymerase chain reaction, that is a PCR, colony hybridization techniques. These all are used for the RDNA technology. So this is the gel electrophoresis, machinery for gel electrophoresis. So this is the phenomena. So here we will apply the mixture of DNA fragments of different sizes, then power on, run the gel, and they may be segregated based upon their size and molecular weight, and then we can see the band of longest and slowest fragments and band of shortest, that is the fastest fragments of the elution, and thereby we will identify the DNA fragments. Blotting techniques, namely southern blotting, northern blotting, and the western blotting, which are extensively used in the RDNA technology. DNA sequencing, there are different DNA sequencing methods like the Sanger's Didiodi method, Maxim Gilbert method, automated method. Artificial gene synthesis is also involved. It is also called as the oligonucleotide synthesis. Dr. Hargovind Khorana and co-workers synthesized artificial gene for tRNA in 1970. Major approaches for artificial gene synthesis are the enzymatic synthesis of gene, chemical synthesis of gene, and the well-known technique, that is the PCR, polymerase chain reaction. So in the PCR, we will go for the amplification of target sequence, and that will be done in the in vitro gene cloning. For the first time, it was introduced in the year of 1985 by Carey Mullis, carried out in the Eppendorf tube. And colony hybridization techniques, there are many in situ hybridization techniques are available. And the next one is the applications of RDNA technology, very very important. RDNA technology used in the different fields. Let us see one by one. Application of RDNA technology in the agriculture. RDNA technology is used to produce transgenic plants. RDNA technology is used to produce transgenic plants. By utilizing tools and techniques of genetic engineering, it is possible to produce genetically modified plants. Many transgenic plants have been developed with better qualities like resistance to herbicides, insects, or viruses. They also allow production of commercially important biochemical compounds, increase nutritional values, increase resistance to stress-free plant. And transgenic plants include the Bt cotton, Bacillus thuringiensis cotton. The insertion of Bt gene into cotton plant has reduced use of the pesticides to kill the caterpillars, which decrease the environment pollution and reduces pesticide resistance in caterpillars. The gene Bt is originally isolated from soil bacterium Bacillus thuringiensis. See here we are forming the Bt crops. The plasmid is removed from the bacterium and the tDNA is cut by restriction enzyme. Second step is foreign DNA is cut by the same enzyme. Then the foreign DNA is cut by the same enzyme. Then the foreign DNA is inserted into the DNA of the plasmid. Then the plasmid is reinserted into the bacterium. Then the bacterium is used to insert the tDNA carrying the foreign gene into the chromosome of a plant cell. Then the plant cells are grown in the culture medium, and finally a plant is generated from a cell clone. All of its cells carry the foreign gene and may express it as a new trait. So thereby the newly found plants, they have the gene of the Bacillus thuringiensis, so that they are called as the Bt crops. They are called as the Bt crops. So there are different kinds of Bt crops which are commercially available. They are Bt corn, Bt rice, Bt cotton, Bt tomato, Bt brinjal, Bt soybean, and Bt potato. And applications of RDNA technology in medicine. Then the next example is the flavor saver tomato. It is the first approved genetically modified food developed by the Colgene in 1922, licensed in May 17th, 1994, ripening of tomato causes production of an enzyme polygalactouredoase, that is PG, in gradual increasing level, which is responsible for the softening of tomato which cause rottening. Hence, collagen is introduced, a gene in plant which synthesizes a complementary mRNA to PG gene and inhibiting the synthesis of PG enzyme. So this is another invention, that is a flavor saver tomato. And coming to the next field, application of RDNA technology in medicine. Recombinant DNA technology is used to prepare vaccines, interferons, and as well as the hormones, monoclonal antibodies, and different kinds of therapeutic agents. This is extensively used in the preparation of vaccines. What are the vaccines? Vaccines are the chemical preparations containing a pathogen in attenuated or inactive state that may be given to human beings or animals to confer immunity to infection. Vaccines are effective against infections used by bacteria or viruses or protozoa. Some of these include vaccines for polio, malaria, cholera, hepatitis, rabies, smallpox, and the recently COVID-19, etc. There are different kinds of vaccines: attenuated whole agent vaccine, inactivated whole agent vaccine, toxoid vaccines, component vaccines or subunit vaccines, conjugated vaccine, recombinant vector vaccine, DNA vaccine, and so on. Here we will see the production of recombinant hepatitis B vaccine. So here we have taken hepatitis B virus, hepatitis B antigen producing gene, and here we have taken out the plasmid, cut the plasmid with the restriction endonuclease enzyme, form a RDNA, introduce into a host cell and go for the amplification. So here we will find out the HB antigen and extract and purify the HB vaccine, and that will be ready for use in the hospitals and clinics. And production of insulin in the form of hormones. Here, by advent techniques of RDNA technology, many hormones like the insulin, somatostatin, somatotropin are produced. Insulin is a hormone produced in pancreas by islets of Langerhans. It regulates amount of glucose in blood. Malfunction of insulin production leads to a condition called diabetes. Human insulin contains two chains, A and B. Chain A has 21 amino acids and chain B has 30 amino acids. Both chains are separately produced as proinsulin and modified to produce proinsulin by combining chains. Proinsulin, when attached to a signal molecule, becomes a functional insulin. There are different methods to produce the insulin. Here we have taken out the plasmid DNA cut with the restriction endonuclease enzyme. Here there is a human cell and human insulin producing gene. We form a recombinant DNA and introduce into the host, and that is engineered to bacteria to multiply the producing insulin, and the insulin is separated, purified to produce human insulin, and that will be injected into the diabetes patients. So there are different methods are employed to produce the insulin in the laboratories. And human growth hormone HGH, and production of transgenic animals. Production of transgenic animals is also the application of RDNA technology. The method of recombinant DNA technology aids the animal breeders to increase the speed and range of selective breeding in case of animals. Important use of transgenic animals are for the production of certain proteins and pharmaceutical compounds. Biotechnologists have successfully produced transgenic mice used to study human immune system, chickens more resistant to infection, cows increase milk supply. Goats, sheep, pigs produce human proteins in their milk. Here, the transgenic animal is one that carries foreign gene which is inserted into the genome. First discovered transgenic animal is the mice, used to study human immune system. This is most widely used animal to detect gene expression, used in knockout technology to detect gene function. And Alzheimer mouse, in the brain of Alzheimer's patient, dead nerve cells are entangled in a protein called amyloid. Mouse made by introducing amyloid precursor gene into fertilized egg of mice. Onco mouse, it serves as mouse model to study cancer. It is made by inserting activated oncogenes. Smart mouse, it is a biological model engineered to overexpress NR3B receptor in synaptic pathway. This makes the mice learn faster like juveniles throughout their lives. And there are chickens which are more resistant to infection, and then cows, they increase the milk supply. Cattle, that is transgenic cows, are made to produce proteins lactoferrin and interferons in their milk. Prions free cows resistance to mad cow disease. Goats, sheep, pigs produce human proteins in their milk. So here transgenic mice, transgenic rabbit is also there. Alba, the EGFP, enhanced green fluorescent protein, created in 2000. Transgenic monkey, Andy, born in 2000. Transgenic fish, they are the glowfish and superfish. The superfish is the transgenic salmon, grows of about 10 to 11 times faster than normal fish. Growth hormone gene is inserted into the fertilized egg. It increases growth and increase in the growth and size. Glowfish, it is a genetically modified freshwater zebrafish. Danio rario is produced by integrating a fluorescent protein gene from jellyfish into embryo of fish. So this is the dolly production. It is the SCNT technique, somatic cell nuclear transfer technique. And transgenic goats are also there. Goats could express tissue plasminogen activator, antithrombin 3, spider silk, etc. in their milk. And applications of RDNA in the industry. Recombinant DNA technology also plays a major role in industrial production, which includes fermentation technology, antibiotic preparation, production of alcoholic beverages, production of vitamins and amino acids, production of enzymes. So this is the utilization of RDNA technology in the industrial area. Most of the times in the industry, microorganisms are extensively used. As per the microbiology, and microorganisms are considered as a miniature chemical factories. They have the capacity to convert a raw material into useful and valuable end product. Industrial microbiology has experienced a dramatic expansion after the invention of the recombinant DNA technology, which helped in producing new genetically engineered microorganisms. The important microbial products are the microbial cells that are used as the immunizing agents, as the vaccines and food supplements. The large molecules like the enzymes, primary metabolic products, that is products that are used by microorganisms like vitamins. Secondary metabolic products, that is products that are not used by microbes like antibiotics. There are in the fermentation technology, here we will see the ethanol, beverages, distilled beverages, carbonic acids, and the vitamin B12. If we see the ethyl alcohol is a common solvent used in producing gasohol, that is alcohol and gasoline mixture used as a fuel. Cornstarch, molasses, sugar beets, potatoes, and grapes are the raw materials from which ethanol is produced industrially by using the yeast Saccharomyces cerevisiae. Coming to the beverages, beverages like whiskey, brandy, rum, beer, wine, and shake are produced using the microbes. Organic acids like vinegar, citric acid, lactic acid, amino acids, and vitamins are also produced industrially. Commercial vinegar, that is 3 to 5% of acetic acid, is produced by using yeast Saccharomyces cerevisiae and bacterium Acetobacter from cornstarch, molasses, etc. Aspergillus niger is used to produce citric acid from cornstarch. Lactobacillus bulgaricus is used to produce lactic acid from molasses and dairy whey. Micrococcus glutamicus is used to produce amino acid glutamic acid from alpha ketoglutarate acid of citric acid cycle. Vitamin B2 and B12 are used industrially produced by using microbes like Ashbia gossypi that produce vitamin B2 and Streptomyces olivaceus vitamin B12. So this is the use of RDNA technology in the fermentation technology. Coming to the antibiotics, an antibiotic is a substance that destroys or inhibits the growth of other microorganisms. Alexander Fleming, 1929, observed the antibiotic activity of penicillin produced by the mold Penicillium notatum while working with the bacterium Staphylococcus aureus. In 1945, Fleming, E. Chain, W. Flory developed industrial production of penicillin using Penicillium chrysogenum. Other antibiotics were produced later. Waxman discovered streptomycin from Streptomyces griseus. These antibiotics prevent cell wall synthesis or damage the cytoplasmic membrane or inhibit nucleic acid synthesis and protein synthesis in microorganisms. Penicillin, cephalosporin, gentamicin, kanamycin, erythromycin, chloramphenicol, bacitracin, cholesterol are antibacterial. Nystatin, griseofulvin are the antifungal. There are different kinds of probiotics are also. Lactobacilli and bifidobacteria are mainly used as the probiotics. Nowadays, probiotics are also used to remove the harmful competitors from the body. So these are the different antibiotics. Coming to the enzymes, enzymes are the biological catalysts. Biological source of enzymes are animals, plants, and microorganisms. Enzymes of microbes are produced commercially by using some microorganisms. For example, enzyme streptokinase, which is used in dissolving the blocks of coronary and cerebral blood vessels to prevent attacks or strokes, is produced by the bacterium Streptococcus pyogenes. The DNA polymerase produced by the Thermus aquaticus, which is called as the TAQ, which is extensively used in the polymerase chain reaction PCR. Cellulase produced by the Trichoderma ricci is used in fabric softening and brightening. Alkaline serine protease produced by Bacillus licheniformis is used to remove the protein stains. Renin is industrially produced by using Mucor pusillus and is used in coagulation of milk and of cheese preparation. So, designer enzymes for specific activities are produced using RDNA technology. These enzymes are used in detergents, in manufacturing photographic products and rubber products, and in industries like baking, brewing, dairy, fruit juice, and the paper industry. And just now we discussed the different kinds of vaccines: attenuated, inactivated, whole agent vaccines, taxide vaccines, subunit vaccines, conjugate vaccine, recombinant vector vaccine, and DNA vaccines are also produced by RDNA technology. And the hormones, biotechnology is also used in producing hormones like insulin, human growth hormone, somatostatin, erythropoietin. Just now we discussed the production of the insulin. And applications of RDNA technology in the environment. RDNA technology is used in eradication of pollution, sewage treatment, bioremediation. By recombinant DNA technology, we can remediate environmental pollutants. Some harmful chemicals that are unable to digest and degrade or partially degraded by TNT chemicals. In this nitrogen reacted with oxygen and superoxide is formed, that is toxic. Knock out the monodehydroascorbate reductase gene that will create resistance against the TNT. Arsenic particles are toxic and they are present in the soil, that is harmful for the plants. PVACR3 gene is inserted in Arabidopsis, which possess reductase enzyme that reduce arsenic. This gene expression will give rise to generate tolerance to arsenic. By recombinant DNA technology, plastic degradation can be enhanced. By genetically modified organisms. Genetically modified strain of Pseudomonas putida can degrade chemical in oil spills. Bacteria that have been genetically modified as bioluminescents and give off light in response to chemicals. This is all about the applications of RDNA technology. By this, we will conclude the RDNA technology, principle, procedure, and the applications of RDNA technology. Thank you one and all.